Life began in water more than 3 billion years ago. And it's still the habitat of the largest mammal on our planet, the whale. And yet, despite its size, this huge animal developed from a single microscopic cell. A cell that divided again and again, the fertilized egg cell. This too is a cell, about to divide. What will develop from this cell? Who provides this particular cell with the correct instructions, perhaps for the formation of a mighty redwood? A newborn baby. He too developed from a single fertilized egg cell that underwent an inconceivable number of divisions. How did each one of these resulting cells know whether it would one day become a skin cell, a retina cell in the eye, or a bone cell in a finger of the right hand? Science, in answer to these questions, says that it's genes that determine development. Another example of cell division, the embryonic stage of a fly. Once again, a program is being carried out, but this time something seems to have got mixed up. Legs at the head, where feelers should be. What went wrong here? And why has the spinal column in this human fetus split into 2 branches, each one with a head? What happened to the gene's control function in this case? A last example of dividing cells. But they'll never produce a living creature or an organ. These cells proliferate uncontrollably, spread relentlessly, and become a deadly menace for the healthy cells in their vicinity. These are tumor cells. Cancer. What controls failed to function in this case? What program was rewritten? Were genes involved here too? And could they be the same genes that determine the development of a healthy organism, except that they have been slightly changed? Changed by what? Can the development of life and its destruction be so closely connected? An intriguing thought. Only if we ask such questions will we ever find the answers. Answers which affect the fundamentals of life itself. But just asking questions is not enough. How can they be answered?
Con la ayuda de estas nuevas instalaciones, hemos logrado disminuir.
Natural laws know no language barriers, but scientific research is restricted by financial barriers. Countries without sufficient financial resources quickly fall behind. Without research, the training and instruction of young scientists is impossible. These countries face the danger of being caught in a vicious circle. In an effort to alleviate this situation, 14 European states and Israel have formed a partnership. The embl is a European science centre with its headquarters in Heidelberg, and as such, it has the same importance for molecular biologists as CERN has for particle physicists. Its objectives: fundamental research, teaching and training, and also the development of new technologies in the field of the life sciences, the discipline that will determine the future. All member countries are given the opportunity to swiftly participate in new scientific discoveries and applications. The Swedish Director General of the embl, Lennart Philipson, explains.
The delegation from the 15 member states of the embl, they meet every year to advise on the scientific program and on several administrative questions, and of course to discuss the financial questions at the embl. After all, financing is not the most important part of the embl program. It's even more important to identify the intellectual resources. In fact, one of our major contributions on the European scene is to train young students into top-class independent scientists. The average age of our 320 scientists is therefore only around 30 years.
Back to the living cell, and to the people who, since the invention of the microscope, have been fascinated by this cell. Today, their interest is greater than ever, because in the cell nucleus, are stored precisely those programs that control the cycle of growth and decay in life, the chromosomes. These are briefly visible during cell division, when they appear vague, blurred, as if in a fog. Seeing more is knowing more. But is it possible to see in a fog? Apparently it is, with a microscope specially designed by the embl. The microscope itself seems to have become hardly more than an accessory. Laser beams pass over the cell sample, scanning the desired information, point by point. From the data, a computer constructs images from the interior of the cell, producing sharp and brilliant pictures. It's possible to image any desired section, and for the first time, in 3 dimensions. And far more shows up on 3-dimensional images.
We can now make high contrast images of thick and optical dense specimens. This is the main advantage of these instruments which we developed during the last 8 years. The technical development of such an instrument is only part of a successful story. It is at least as important to work interdisciplinary with specialists from different fields. And now we need biologists who work at the forefront of biological research to make the best use of such a microscope. The structure of the embl is excellently suited for these collaborations.
Cell division, as it has never been seen before. When a cell divides, each of the 2 daughter cells receives the same set of chromosomes, the same hereditary information, the same genes. But where are these genes actually to be found? The chromosome, as a thread with multiple twists and coils. Its individual segments. These are the coded building instructions for proteins. For example, those proteins that give our hair its particular quality and color. Or instructions for those proteins that lend our skin its typical features. Our body consists of 100,000 different proteins that cause it to function. Proteins that require 100,000 building instructions, precisely those segments we saw on the chromosome thread, in other words, the genes. The relationship between genes and proteins is like that between an alphabet and a language. Each gene is a precisely defined sequence of chemical building blocks, the letters of the genetic alphabet, used for writing the building instructions on the DNA helix. 10 centimeters of DNA with about 5,000 building instructions, coiled and twisted to form a chromosome that is 10,000 times smaller. And there are almost 50 of these chromosomes in the nucleus of each body cell. Oddly enough, little is to be seen of these chromosomes after cell division. Does that mean that their valuable information has also been lost? Maria Carmo Fonseca from Portugal and Rainer Peppercock from Germany are searching for answers to this question. They believe that when looking for truffles, it's best to rely on the nose of a truffle pig. And this is exactly what they are doing. They inject sensors into the cell nucleus, or more precisely, the injections are performed by a fully automatic, extremely accurate device that was specially developed at the EMBL. This patented device is being used in scientific institutes around the world. The cell nucleus no longer appears to be a structureless mass. On the contrary. Another member of the team is Angus Lamond from Scotland.
Cells store their genetic information in the form of genes, which are kept within the nucleus. In order for this information to be put to work, a message has to be sent from the genes out of the nucleus. This message then instructs the cells how to build the proteins they need to live and grow. We're particularly interested to understand just how these messages are produced and sent out of the nucleus.
Using specific probes, we are now able to start localizing where within the nucleus these different processes take place. In particular, we are studying the function of a novel and specific compartment, which will be seen in these pictures as bright spots.
A first glimpse into a world which until now was largely impenetrable. But how is it possible to render visible something that will never be visible? In other words, the molecule itself. A segment of the DNA helix. Illuminated here, the beginning of a particular gene, a sort of chapter heading in the instruction manual of the cell.
Okay, so I can see them both.
The helices.
A protein approaches, attracted by the starting mark of the gene as if by magnetic force.
The DNA is also rotating around.
This is the typical binding.
The protein docks on the DNA.
Yeah, but it's
It constitutes a sort of gene switch that triggers the subsequent copying process.
It does show the helix fits in very well.
How are computer images of this sort produced? Is this, in fact, reality? The gene and its switch in an aqueous solution as they occur in the cell. But here, they're expected to crystallize, to form a solid crystal structure. Frequently, it takes weeks to acquire the knack and for the desired crystals to appear. Often, it just doesn't work. If it is successful, then it's a matter of routine. The X-ray analyzer runs day and night and precisely determines the position of each single atom. The structure of molecules determines their function.
Somewhere in here should be the DNA. The question is where? This is an obvious channel here, but there doesn't seem to be Yeah, the protein is strong.
And that's the gap there is where...
To elucidate the interactions between the genes and their own products, the proteins, requires collaboration between various specialized disciplines: microbiology, biochemistry, physics, and computer technology.
So this doesn't open the minor groove?
These two oxygens. Yeah.
That's the arginine.
A lot of patience, a lot of hard work, as well as luck, is required to accomplish another small step forward.
So we've...
Like here,
So which
how a
axis are we looking
protein
at?
recognizes the starting mark of a gene.
Yeah,
So you can see them here.
down the z-axis. Yeah.
Peter went to
But other questions immediately
Here
arise.
the 3-2-4
Maybe the upper one is phosphorylated.
All 100,000 of man's genes in every single one of his body cells?
Yes, it's not
Whereas a fraction of this total information is sufficient to program a cell to function as a retina cell in the eye, another fraction contains the building instructions for the muscle cells that move the eyelid.
And I don't understand.
How then does a cell learn whether it will finally become a retina cell or a muscle cell? Who or what finally decides which of the 100,000 genes are to be switched on or become active, and which are not to be switched on, and where this is to occur? Back to the early stages of embryonic development. Here, Denis Dubourg from Switzerland has made some fascinating discoveries in mice embryos. He observed that on the chromosomes, certain genes are switched on only in certain body regions. A gene, here marked in yellow, closer to the beginning of the chromosome, is switched on from the 6th cervical vertebra onwards. While this gene, marked in blue, closer to the end of the chromosome, is only activated from the 9th vertebra, that is, more towards the end of the body. Further genes of this kind were found. The closer they were to the beginning of the chromosome, the closer the related body zone was to the head. The closer they were to the end of the chromosome, the further the related body zone was away from the head. The order of these genes along the chromosome axis therefore corresponds exactly with the arrangement of limbs and organs along the body axis, from head to tail. Are these genes actually master genes, which transmit instructions that are specific to particular body regions? Some years ago, similar genes were discovered in the fruit fly. And there too, they control precisely defined body regions. And now, an astonishing discovery. In both the fly and the mouse, subsections of corresponding genes are almost completely identical. Why this molecular kinship? All these gene areas that are almost identical are the programs for protein areas, which in principle are very similar to the gene switches that we've already met. They dock on the starting mark on the DNA, and in doing so switch on specific genes. They are thus gene switches, gene activators that control cell growth. They transmit to the cells located in different body regions the instructions for correct behavior in these places, so that, for example, arms grow at the right places and legs grow at other places. Flies and mice are not alone in possessing almost identical gene switches of this sort. The same is true of all living things that have a head and a tail, that is, a body axis. The principle of controlling body development with the aid of gene switches has evidently proven so successful that it's been retained since the dawn of evolution. However, This is not surprising. The genetic alphabet was only invented once. It's valid for all living creatures. All living creatures understand it. Communication on the molecular level, communication on the scientific level. The annual symposium at the EMBL. These scientists from all parts of the world have gathered to concentrate for several days on one important subject.
One for ATP and one for magnesium ADP. What is the evidence for it?
So the evidence that I didn't have the time to present is the following one.
To listen, to be inspired, and to encourage others to adopt new, still unfamiliar ideas and to combine them with their own. The EMBL brings together the molecular biologists of Europe and at the same time, by means of such events, provides them with the opportunity to establish contact and enter into competition with the worldwide research in their field. Since research at the EMBL is financed exclusively by the member states, it's free from application-oriented pressure and free from the obligation of secrecy. Freedom and spontaneity as the essential prerequisites for fundamental research. The decisive discoveries of pure research occur spontaneously. They can't be planned. One of these scientific surprises, oncogenes. The causes for cancer are established within ourselves, in our genes. The very genes that control growth and development function like a boomerang. They run amok. They destroy life. Cancer caused by oncogenes. To speak on this topic, here's Thomas Graf, head of the differentiation program at the EMBL.
Today there are about 100 genes which are known to contribute to cancer and which are commonly called oncogenes. These genes are very highly conserved during evolution and are almost identical between flies and men, for example. Normally these genes are only active when the cell needs them, when the cell should divide. And so they are tightly controlled. But the genes can also be changed, and this can occur, for example, by outside factors like irradiation or chemical substances, in which case these genes can become constitutively active or hyperactive. They are always on, and this of course can lead to a situation where the cells don't stop dividing and can form a cancer, a tumor. In addition to these positively acting genes, there are a number of genes which are now known to counteract growth, which sort of serve as brakes, which are important if a cell wants to stop dividing. And if such genes are destroyed by similar agents as the ones I mentioned before, that can also contribute to cancer. Another important realization has come with discoveries in the last few years, which has shown that the same types of genes, both oncogenes and anti-oncogenes, can also have important functions as master genes in the specialization of cells during embryonal development. And this link has also been one of the reasons why we have recently expanded at the EMBL in this area. We have attracted several groups working on embryonal development, and I think it's also a nice example which shows how fast we can react with the structure here at the EMBL to exciting new developments by attracting group leaders who do interesting work in these new areas.
Growth genes, oncogenes, anti-oncogenes. The words could belong to a magic spell that rewrites the oracle of life. Life is growth and multiplication. Growth and multiplication lead to death. Gene activators control division and multiplication of the cells and lead to the development of life. growth-inhibiting genes counteract them and keep excessive growth in check, a finely tuned balance. If a cell dies, it's replaced by a new one, no more and no less. On the other hand, if the growth genes become overactive, damaged, for example, by environmental influences, or if growth-inhibiting genes fail for the same reason, then life degenerates into deadly growth. The balance is upset. And if the growth-inhibiting genes finally gain the upper hand, this leads to natural aging and also to death. Life is balance. What is creativity in science? What is innovation? Everyone has a different outlook. Creativity means to reach a standpoint, a point of view that unites different outlooks, that makes connections visible. From this standpoint, one perceives that valleys that seem to exist in isolation are in fact connected by their rivers which flow together somewhere. Scientific creativity cannot be programmed. It can be promoted. The EMBL provides the perfect conditions. EMBL, in the heart of Europe, in a region with a renowned scientific tradition and rich cultural heritage.